So thank you for that introduction. Thank you for the invitation to speak today. I'm going to talk today about the evaluation of germline-driven non-genetic inheritance in human phenotypes and diseases. So this is a big topic to get through in 20 minutes with many critical caveats. And I've done my best really to narrow it down to the most important and interesting components. This is a field, as everyone's probably aware, that is still very much developing. There's many more questions than answers in this field, and exactly how what I'm going to discuss fits into the biology of disease inheritance in humans largely remains to be determined. So for about half of this talk, I'm going to discuss the challenges of examining this process in humans, and in the other half, I'm going to discuss some of the most interesting examples from the literature. So before I start, as I'm sure many members of the audience are already aware, this is a very controversial topic for a number of reasons. One of which is that there's significant doubt about whether this phenomenon even really truly exists in humans. This is largely because of the erasure of epigenetic marks that characterize human development soon after fertilization. There's also controversy relating to larger societal implications that come from the realization that our lifestyle decisions, what we drink, eat, medications, toxins, environmental exposures, can really have a significant influence on our children, grandchildren, and possibly even beyond that. So before I get to humans, I want to do a quick review of how this mechanism of inheritance has been examined in mouse models, which will provide a good model as a foundation system that I will then use to compare to how this phenomenon has been evaluated in humans and why examination of this process in humans is much more complicated. So as a brief summary, there's very convincing evidence in mice that various exposures in ancestors can cause stable heritable defects in epigenetic factors in the germ cells, ultimately manifesting as an abnormal phenotype in descendants. A variety of exposures have been well characterized, including high-fat diet, various forms of stress, toxins, and different medications, as well as a bunch of other exposures that I'm not mentioning here. These exposures have been shown to cause stable heritable changes in the molecular composition of germ cells, including DNA methylation marks, as well as the expression of small RNAs, especially Pee-wee RNAs, tRNA fragments, and microRNAs, and so on. These exposed mice then produce offspring that have an increased risk of certain disease phenotypes, such as various metabolic, neurodevelopmental, psychiatric, anatomic, and reproductive diseases. So in many of these experimental systems, the mechanism of inheritance has been narrowed down to the germ cells by use of in vitro fertilization using a sperm or oocyte from an exposed individual, which is then fertilized by a gamete of a non-exposed individual and implanted into a non-exposed female. When this is done, the phenotypes that have been observed are often recapitulated, which really narrows this, this locus of inheritance down to the germline. This approach eliminates potential confounders that we encounter often from the maternal side, including fetal-maternal communication of factors across the placenta, as well as postnatal nurturing and maternal behavior effects. Going one step even further, in some cases, small RNAs, when isolated from exposed germ cells and injected into a wild-type embryo or zygote, can recapitulate the phenotype, further narrowing down the locus of inheritance to specific molecules in the germ cells. So now switching from these well-designed experiments in which the only difference between the exposed and unexposed individuals is the exposure itself, I'm now going to discuss how this phenomenon has been evaluated in humans, which is much more indirect and, to be frank, much more messy. So before I get to that, why is this form of inheritance potentially relevant to human diseases at all? Well, over the past 2 decades, we've made really remarkable progress in evaluating the human genome with tremendous sophistication and in great detail. During this time, the search for genetic variants to explain disease heritability has been largely disappointing, and much of the heritability of common diseases remains unexplained. So in addition to my interest in this field, I'm also a neonatologist and a pediatrician. And there are many pediatric diseases which I see frequently for which the inherited driver of disease remains unknown. So here's a list of some pediatric diseases for which we've largely been unable to find the causative heritable factor. This is not a comprehensive list, but it does include autism, congenital heart disorders, which I'll touch on in a little bit in more detail, pyloric stenosis, which people may not be familiar with, but it's basically a very interesting disorder that is a muscular overgrowth of the intestines that prevents food from being able to move through. And it shows a very interesting pattern of inheritance in which the babies most affected are firstborn sons. This list rounds out with asthma and a variety of behavioral problems, including anxiety, depression, and hyperactivity. So I've expanded this data a little bit here for congenital heart disease, which is something I see frequently in the NICU and often involves multiple very serious surgeries, and often stays in the hospital for months, if not years. So as you can see, the risk of developing congenital heart disease is significantly greater if you have a relative with a history of congenital heart disease. So if you have a first-degree relative, an individual's risk is 3.2-fold greater. If for a second-degree relative, your risk is 1.8-fold greater, with a mild increase in risk for third-degree relatives. Now, in these people, in these families that have congenital heart disease that runs through the families, we've only really been able to localize this heritability to abnormal DNA variants in about 1/3 of the cases. The search for causative DNA mutations has come up empty for the remaining 2/3 of these cases of congenital heart disease that runs in families. So to me, the conclusion from this is that it seems that the search for causative inherited factors should really stretch beyond looking at the DNA to include the many other factors that we know are inherited at the time of fertilization. So compared to what I discussed 2 slides ago in mice, in humans, this connection from exposure to germ cell molecular change, ultimately manifesting in a phenotypic abnormality, especially risk of disease, is much more difficult to establish. And to my knowledge, no single study in humans has connected exposures with change in the heritable content of the germ cells, ultimately leading to a modified risk of disease. However, there have been many studies connecting 2 of these events. For example, we have multiple examples of how different exposures are associated with changes in the profile of DNA methylation marks or small RNA expression. In addition, specific exposures in ancestors have been connected with specific phenotypes in descendants, including children, grandchildren, and even great-grandchildren. Furthermore, to my knowledge, there's been 1 study examining prospectively the association between germ cell epigenetic abnormalities and the risk of disease in offspring. So I'm now going to move on and discuss how these associations have been measured in humans, discussing the challenges in performing these studies, as well as the potential confounders that we really have to consider when trying to draw conclusions regarding mechanisms of inheritance. So, detecting an association between ancestral exposures and phenotype or risk of disease in descendants is very challenging for multiple reasons. For one, the time period between exposure in an ancestor and the development of disease in descendants is very long, often decades. If the exposure occurred during childhood and the disease doesn't manifest until adulthood in the offspring, this period can be 60 years or more. And this is only considering transmission across 1 generation. It's even longer if we consider transmission to grandchildren or great-grandchildren. And characterizing the specific exposures of an individual from 60+ years ago is very challenging. This includes any potential medications. I can speak from personal experience. Seeing medical records of medications from decades in the past is all but impossible here in the US, but also includes unusual environmental exposures such as molds, household chemicals, toxins, abnormalities in the diet. And for some of these, it's even very likely that the person is unaware that they were ever exposed to these factors in the first place. In addition, number 2 down here, many human diseases are complex and seem to be caused by multiple factors, each of which has an incremental additive effect, which makes really discovering one contributor to disease quite difficult. Furthermore, human populations are quite heterogeneous in terms of genetic makeup, but also their various exposures, including diet, smoking, drug use, alcohol consumption, exercise, sleep, medications, toxins, and so on. And this heterogeneity applies to both the ancestors and the descendants. As a result, we really can't compare exposed and non-exposed individuals and assume they are otherwise completely identical, because they almost certainly are not. And trying to draw a conclusion between a single exposure in an ancestor and disease in a descendant becomes very messy. The next few slides, I will discuss 3 specific studies that have shown an association between ancestral exposures and descendants' phenotypes. So probably the most famous example is the Calyx study, which has been described in detail in a number of different places. Basically, by studying the medical records, as well as the records of the quality of a harvest in the small town in northern Sweden over many decades, a clear connection was drawn between the food availability during pre-adolescence, during the ages of about 8 to 12 years, known as the slow growth period, in males, and mortality in their grandsons. More specifically, a low abundance of food around this time was associated with decreased mortality in grandsons, and an overabundance of food was associated with increased mortality. There's a lot more subtlety to these findings involving sex-specific connections and specific types of mortality, which I won't cover here, but the general idea is that grandparental food availability has an influence on the mortality of grandchildren. Importantly, these findings were verified in a number of independent data sets, and in addition, one benefit of this study is the relative homogeneity of the population in this small town in northern Sweden. which may have helped the investigators to detect a signal among all of the noise that I discussed previously. So the next example that I will discuss was only recently published, and it looks at the effects of prenatal thyroid hormone exposure on thyroid hormone metabolism in descendants. So inhabitants of the small island known as São Miguel off of Portugal are high-frequency heterozygous carriers of an autosomal dominant thyroid hormone receptor mutation that causes decreased sensitivity to thyroid hormone. The carriers of this mutation compensate for this decreased sensitivity by generating higher levels of circulating thyroid hormone. An important point here is that this is a compensated hyperthyroid state. And what I mean by that is that these people have high thyroid hormone levels, but because of the decreased sensitivity of the receptor, they don't suffer any sequelae of hyperthyroid disease that we commonly worry about, such as metabolic, kidney, cardiac, or any other end organ disease. In my opinion, this is really an excellent model to study the effects of exposures across generations. In this case, the exposure is high circulating levels of thyroid hormone in utero during fetal development, and the authors examined how this affects descendants who did not inherit the mutation and therefore are not genetically predisposed to abnormal thyroid hormone metabolism. An important point here is that the F1, F2, and F3 generations are all wild type. There was exposure to elevated thyroid hormone in utero in the F1 fetus, as well as the germ cells that eventually formed the F2 individuals. On the other hand, the F3 generation was never directly exposed to elevated thyroid hormone. The phenotype that the investigators analyzed is known as reduced sensitivity to thyroid hormone. This is a little complicated, but basically what it means is that when given exogenous thyroid hormone, someone with reduced sensitivity to thyroid hormone shows a failure to inhibit thyroid hormone release in response to subsequent challenge or stimulation with a thyroid stimulating hormone. Very complicated. For the sake of this presentation, we can just consider these people to have abnormal thyroid hormone metabolism. So when the investigators looked at the F1 generation, not surprisingly, they saw reduced sensitivity to thyroid hormone and also low birth weight. All of this is very consistent with exposure to high levels of thyroid hormone in utero. Intriguingly, when they looked at the F2 generation, which we can see here, the children of F1 males show evidence of reduced sensitivity to thyroid hormone, but the children of F1 females do not. Moving along to the F3 generation, which was never directly exposed to thyroid hormone in utero, children of F2 males show reduced sensitivity to thyroid hormone, similar to what was observed in the F2 generation. However, there was no effect observed in the F3 children of females. Thus, these F3 males and females display altered thyroid hormone metabolism that has been inherited down 3 generations of wild-type individuals in people who were never directly exposed to high thyroid hormone metabolism. So the benefit of this study is that the exposure is relatively homogeneous, and the phenotypic readout, this reduced sensitivity to thyroid hormone, is very specific and very easily quantified, which allows for a very clear association to be drawn across multiple generations. I just wanted to give a plug that Arturo Hernandez, who will be speaking in a future session, has demonstrated a similar effect in mice, albeit looking at a different mutation and analyzing effects on behavior and gene expression in the brain in the F2 and F3 generations. So the next association I will mention only briefly, but that is that there are a number of studies showing a connection between smoking while pregnant in a female and the risk of asthma in her grandchildren. This effect shows a dose responsiveness such that asthma was more common in the grandchildren of women who smoked more cigarettes per day. And importantly, these connections persisted after adjusting for a lot of variables, many of the variables that we have to consider that I mentioned in previous slides. One of the most important ones being maternal smoking. So this effect still persisted even when accounting for the amount of maternal smoking. There are many more studies demonstrating an association between exposure in an ancestor and a risk of disease in descendants that I will not discuss here, but I just wanted to stress that these associations have been observed in multiple other studies, some of which are listed in this table below, which comes from this review that Jill referenced that I recently published with my fellow Liana Sinaldi. So these phenotypes that I've just discussed are potentially caused by the inheritance of abnormal germline molecules. However, non-germline mediated mechanisms of inheritance are also possible and really must be considered. We have to keep in mind that exposures have wide-ranging effects beyond just the germline, and they can affect one's life trajectory in a variety of ways and affect inheritance of phenotypes completely independent of any germline effect. For example, parental stress is a very commonly tested exposure. However, in addition to any possible effects on the germline, stress also affects one's parenting style and can affect one's offsprings in a variety of different ways that can have a significant effect on their health. Also, I just wanted to mention that for many of these studies, the locus of inheritance has not been localized to the germline as it has in mice with the in vitro fertilization experiments, as I've mentioned. And contributions from fetal-maternal communication across the placenta or postnatal maternal care are also possible and really must be considered, especially when looking at inheritance down the maternal lines. So I'll now quickly discuss a few select studies that show a connection between an exposure in an individual and specific epigenetic abnormalities in their germ cells. For pragmatic reasons, almost all of this work has been performed in sperm. So first thing I want to mention is that Roman Barris's group has shown that people who undergo 6 weeks of endurance training develop changes in DNA methylation and small RNA expression, especially involving Peewee RNAs, and gene ontology analysis of these differentially methylated regions revealed enrichment for genes that regulate neurologic function. In addition, childhood abuse has been associated with significant changes in sperm DNA methylation, especially around genes that regulate neuronal function of the immune system. I don't want to steal anyone's thunder with the next association. People in the next few sessions are going to be discussing these, but smoking both cigarettes and cannabis has been associated with sperm epigenetic abnormalities, including microRNA expression and DNA methylation. Finally, exposure to a variety of chemicals, including mercury, Agent Orange, bisphenol A, chemotherapeutics, have been associated with alterations in the DNA methylation in sperm. And I think some of our speakers today and in future sessions are going to touch on some of these agents. So similar to what I discussed previously regarding confounders when searching for connections between ancestral exposures and diseases in descendants, we really must be careful when drawing associations between exposures in an individual and changes to the molecular profile of their germ cells. For example, we have to be very cautious not to assign causation or attribute observed differences to the exposure that we've decided to evaluate. Certainly those who were exposed differ from the non-exposed people in more parameters than just the exposure itself. This includes factors that make it more likely for someone to be exposed, as well as consequences of the exposure itself, both of which have an impact on offspring phenotypes independent of that exposure. A second critical point is that, as I'm sure many people in the audience are aware, not all differences in DNA methylation or small RNA expression are biologically meaningful. Some differences are statistically significant, but so small that they're unlikely to have biologic significance or biologic meaning. Similarly, the significance of some DNA methylation abnormalities in gene-poor regions of the genome have unclear relevance to disease inheritance. Finally, I'm going to discuss the one study that I'm aware of that prospectively evaluated a connection between parental germ cell epigenetic abnormalities and disease in offspring, specifically talking about diseases in offspring that are not canonical epigenetic diseases. For example, obviously imprinting disorders are from inherited epigenetic defects, but these are well-defined epigenetic disorders. What I'm referring more to is common complex human diseases. And so this study looked at DNA methylation in fathers and the development of autism phenotypes in the offspring. To do this, the investigators utilized a cohort of fathers that already have a child with autism and were therefore determined to be high risk to have another child with autism. During a subsequent pregnancy, these males, who were identified as being high risk because they have a previous child with autism, were contacted and they submitted a sperm sample for DNA methylation analysis. Once born, that infant was then monitored for symptoms of autism. So when this study was performed, the investigators found regions of altered DNA methylation in fathers that were associated with infant autism phenotypes, especially around genes involved in developmental processes. One thing I really wanted to stress is that this type of study, in my opinion, is very valuable in understanding the germline non-DNA contributions to diseases in humans. And I think a wider application of this approach moving forward will provide tremendous insight. So borrowing from this prospective approach, future examinations may really benefit from starting with a disease in which the search for causative factors in DNA sequence has not explained the observed heritability. A good example would be congenital cardiac disease, as I mentioned earlier, which enriches in families and we've been unable to find largely the inherited driving factor. And by focusing on families that have multiple children affected by congenital cardiac disease, the parental germ cells can then be examined for abnormalities in DNA methylation, small RNA expression, etc. By focusing on these enriched cohorts in which there's multiple cases of disease in one family, the chances of detecting a true signal are much greater. In addition, this approach removes a lot of the noise encountered when trying to compare exposed with non-exposed individuals. Of course, there's caveats to this approach that must be considered. First, the epigenetic profile of germ cells at the time of measurement may be quite different from it was at the time of fertilization. Some of these modifications, while being quite sensitive to the environment, are also very dynamic, and between weeks or months, they can change pretty significantly over time, especially in sperm. In addition, all of this work is really currently much more pragmatic in the paternal germline for the relative ease of obtaining sperm compared to oocytes from females. However, the benefit of this approach is that if common defects are detected in germ cells, then these differences can be investigated for a common exposure that can explain the differences. In this sense, starting with the disease and working backwards towards the exposure may be a more focused and much more efficient approach to find the true causation of disease. This approach can also be performed by taking individuals with a specific disease and looking retrospectively for common exposures in their parents, very similar to what Jill Escher is doing with autism and parental exposure to general anesthesia. With that, I just wanted to thank Jill for the invitation to give this presentation today, as well as my colleagues and all the members of my lab at Cornell. Thank you very much.
Thank you so much, Dr. Smith-Raska. That was a great summary. And you're right, it's very hard to do. I mean, you could have done half an hour just on DES, but I did want to point out to everybody listening that our next 3 sessions do touch on, I'll do my video, do touch on evidence in humans in endocrine disruption in tobacco and cannabis, and also not in general anesthesia, because there's never been a human study, but hopefully there will be, yes. Just like you pointed out. So I don't see any questions from our audience, but oh, sorry, go ahead, Tia.
Oh, yes, I was going to say, so I have a question about one of the studies that you had discussed. Yes. It was with the thyroid hormone and looking at the multiple generations and looking to see that only males were affected. My question was, is it only
children of males, not children. So males and females were affected, but only the children of males. So presumably, yes, it's passed down through the maternal line, but not through the maternal line.
Okay, got it. So my question was, one of the health outcomes that was seen in the offspring, was it only the low birth weights, or was the study followed up until they grew older for their long-term adult health outcomes?
So the low birth weight was just in the first generation, the F1, and that's probably just because of the direct effect of thyroid hormone on the developing fetus. But so low birth weight, but then the reduced sensitivity of thyroid hormone, the thyroid hormone metabolism defect, was actually measured in adults. So these people were followed throughout their life. And this is they're not really disease. This is just an interesting phenotype you find in a response to exogenous thyroid hormone administration. So I wouldn't say that this is a disease that tracks through time, but an evidence of a phenotype, essentially, a metabolic abnormality that persisted down generations, basically independent of the ancestral mutation and also independent of any in utero exposures, which we saw in the F3 generation.
Okay. All right. Perfect. All right.
Thank you. So Tia has a couple of changes in our lineup for the short talks. Thank you so much, Dr. Smith-Raska.
Yes,
appreciate it.
Thank you, Matthew.
Yep, my pleasure.